Good evening, everyone. My name is Jason Anderson, and my project is on the effect of interventricular dyssynchrony on myocardial strain. Now, before I get into all the jargon in my title, I'll really explain why I wanted to do this. Well, first, my grandfather had aortic valve replacement surgery when I was in the 6th grade, and more recently, one of my close friends was diagnosed with mild hypertrophic cardiomyopathy. And all of this piqued my interest in the field of cardiology. I was fortunate enough to get an internship at OHSU, and even more fortunate to find a lab that gave me the freedom to research what I found interesting. And that was interventricular dyssynchrony. Now, what is interventricular dyssynchrony? It's two really long words that pretty much just means there's a difference in timing between the left and right ventricles. Now, it's a manifestation of congestive heart failure. Congestive heart failure as well as heart disease are becoming more prevalent in the United States and other developed countries. And one of the issues right now is that there aren't very many treatments for mechanical dyssynchrony. And a method of treatment that's been developed in the last 5 to 6 years is called cardiac resynchronization therapy, or CRT. And this is a new method of pacing that is quite different from a traditional pacemaker. Now, a traditional pacemaker replaces the SA node and basically just controls the standard heartbeats and acts to start the heart's contraction, whereas CRT acts as sort of a modified defibrillator, where it actually has nodes, leads connected to the SAAV nodes and the Purkinje fibers that depolarize the heart, then shock it back into working. And this corrects major arrhythmias such as interventricular dyssynchrony and other mechanical and electrical dyssynchronies. Now, a major issue with CRT is that in current clinical studies, there is very little accuracy in terms of predicting whether CRT will work or not. And in reading studies, I realized that there weren't any in vitro models that simulate dyssynchrony, so that scientists and other researchers could figure out how to improve CRT to get the accuracy up. And so my goal was to create a model, an in vitro model, that simulates dyssynchrony and understands how the disease actually impacts cardiac performance. Now, looking at some basic cardiac anatomy, as you can see, there's a spiral shape to the tissue. This creates lateral tissue movement during the cardiac cycle. And what this does is makes the heart beat as if you're wringing out a towel. It creates twist as well as compaction of the heart. Now, what this does is create something called strain. And now, many of you may think strain has a very negative connotation in the English language, but all it is a vector that measures the percent change in tissue movement. Now, here's the fundamentals of strain. You see this. Imagine this is the heart, and see this point here. When it's contracting, this point moves in, and the change in position, that percentage, is what strain is. And so this laterally is circumferential. So circumferential strain is what I'm measuring is designed to, what is normally is negative because it's contracting in. And this is what my dependent variable will be. And looking at the heart here, you can actually see the heart twisting in this ultrasound image, and that is what creates strain, is that twist. And what you just saw there is something called 4D speckle tracking analysis. Now, speckle tracking analysis is a relatively new method of determining strain. The traditional method was used by something called sonomicrometry. And while sono is very accurate, it requires suturing crystals to the heart itself. And you can't really just cut someone's chest open every time you want to figure out how their heart is doing. And so something like speckle tracking enables doctors to have a non-invasive method of determining an index of cardiac performance. And basically how it works is that ultrasound images are divided into a grid, and then this grid is divided into points called speckles. And then, as the name suggests, the movement of the speckles is tracked, and this is used to calculate strain. Now, what you saw there was 4D, or real-time three-dimensional echocardiography. There are two major types of ultrasound that are used in research right now, 4D and 2D. And the second goal of my study was to look at whether 4D or 2D is more accurate in this case. Now, to my model itself, I used a pig model, and pig hearts are analogous to human hearts in their physiological function, and that's why they're used generally for in vitro studies. And what's really important about my project is that it's the first successful demonstration of dyssynchrony in an in vitro model that quantifies the dyssynchrony itself. Previous studies have simply shown, yes, there is dyssynchrony, there's not dyssynchrony, how does this change the heart? Whereas my study actually looks at how much there is and incrementally increases the amount of dyssynchrony and see how that affects cardiac performance. And this leads me to my hypothesis, which is my independent variable is the milliseconds of dyssynchrony between the left and right ventricle. And my hypothesis is as interventricular dyssynchrony increases, there will be significant decrease in circumferential strain in porcine hearts. Now, looking at my methods, start out with freshly butchered pig hearts. There is a great day on Tuesday to smell that blood in the lab. And next, the hearts, I then cut and clean the hearts and remove the atria as well as all the other vascular tissue. From there, I spent a long time suturing tubes with balloons connected to them to act as passive models of the ventricles to the annulus of the mitral and tricuspid valves, as well as sutured shut the outflow tracts from the left and right ventricles. And then what was more unique about my project was the model itself. There are very few labs in the United States, as well as around the world, that do passive in vitro models, and all of them use analog pumps like the ones seen here. Therefore, one issue with trying to replicate dyssynchrony is that when you try to turn two things on at the same time, it's really hard to make that accurate. And so what I did was design a system of circuits and a program that was actually able to detect how many milliseconds off the two pumps are each other, and therefore I could dial the two pumps in to where I wanted them. And this is where I spent quite a bit of time, a majority of the time on my project. And as you can see, here is like the actual graph working in one pump, a second pump, some classy Batman duct tape, and the overall experimental model here. And my hearts were imaged underwater because water is what mostly comprises the human body, and this is a very good medium for ultrasound waves to actually pass through. And then just a basic ultrasound machine. And then here, as you can see, are some analysis software. And again, there's a formula. Just remember, it's a % change in tissue position. And here you can see the peaks values, which are the values that I recorded. And these correspond to different peaks, correspond to different segments of the ventricle, as you can see there. And I was looking at the septum, so I record values from this column here. And this is the same sort of thing, except in 4D. Now here is the average septal 4D circumferential strain over 5 levels of dyssynchrony, 0, 20, 40, 60, and 80. And I did this over 8 trials. And as you can see here, there's a significant increase in circumferential strain between 20 and 40 milliseconds. This suggests that somewhere between 20 and 40 milliseconds, there is a significant decrease in cardiac performance. As well as the non-overlapping standard deviation bars suggest that there is low variability within data. Paired t-tests between each of the columns reveal p-values of less than 0.001, which shows that the data is statistically significant. Looking at 2D circumferential strain over the same 5 levels of dyssynchrony, you can see that between 20 and 40 milliseconds this time, there's actually a change in sign, which if you remember, since it's a vector, that means the cardiac tissue is actually moving in the opposite direction of how it should. This means that the cardiac performance is inherently limited somewhere between 20 and 40 milliseconds, which looking at these two graphs, it suggests that 40 milliseconds appears to be the epicenter of where cardiac performance is decreasing. And again, the non-overlap of standard deviation bars illustrates low variability within the data, and again, the paired t-tests reveal all values of less than 0.001. And this can be again seen in the analysis software here, where you have negative peaks on a healthy heart in circumferential strain, and then positive peaks over here on the. And what does this mean for CRT? Well, as I said earlier, congestive heart failure is becoming more prevalent in the United States and other developed countries. We don't have that many effective tools to prevent and fix major issues such as dyssynchrony itself. That is why technology like CRT is really needed in the United States and other countries. Now, the fact that I was able to see such drastic change in strain at 40 milliseconds indicates that there is an actual dip in cardiac performance at this level. Now that leads me to believe that 40 milliseconds is the threshold for where CRT should be implemented by that point. Now, this identification of how the heart function changes can help better develop CRT as well as better develop diagnostics techniques. With a better understanding of how the heart actually changes function, engineers who are developing CRT can better fine-tune the actual electrical impulses to correct the movement of the heart. As well as in terms of diagnosis, ultrasound technicians and other researchers can better know how to use the transducers to actually detect this shift in direction. And looking at 4D versus 2D echocardiography, RT3DE has been widely used in research, but not in clinical practice. It is even used in obstetrics, but not in cardiology. And in my study, I found that both illustrated change, but RT3DE did not show this change in direction. And this is a failure of software. The 4D software that I used displayed the positive peaks on this graph, but since circumferential strain is not supposed to be positive at most times, I was not able to actually select the points manually, and I wasn't able to record that data. As you can see, 80 milliseconds here even gets very close to 0 when it should be over in the positive direction. So with better software, I would be able to actually have more similar data. So looking at 4D as a whole, in theory, it has more accuracy, and as well as in other studies have validated that it has better accuracy in 2D because it doesn't rely on geometric assumptions. So I believe it should be implemented in the future. And for me, future research, I know there is better GE software available, and I'll be working with that over the next summer to help reanalyze my data, as well as analyze the rest of my data for different types of strain and focus more on my research project. I'd like to thank the OHSU Cardiac Fluid Dynamics and Imaging Lab, as well as Drs. David Son, Mohammed Ashraf, and Meiwa Zhu, as well as the lovely Mrs. Kelly Cameron, my teacher and mentor. Thanks. Questions?
So in your ultrasound images, there's color associated. What's going on there? What's the color?
This image, or what exactly?
I guess in kind of near the beginning of the presentation.
The video kind of clip, or okay. So this is a different software. This is actually a Toshiba software that's actually looking at rotation in this graph. So these colors are actually, well, it's Doppler. So then it'd be based on the rotation. You can detect how much the actual heart is twisting. So the different colors represent how much the heart is twisting itself.
For each of the different independent millisecond, we call it Dyssynchrony, how large are your sample sizes?
Okay, so the question was for each level of Dyssynchrony, how large is my sample size? So in the setup, each heart was imaged at each level of Dyssynchrony, and I did the trials over 8 hearts, which is typical. Between 6 to 10 hearts is what's typical for an in vitro passive study. So that I had 8 trials for each of the, each heart was imaged with 3 trials per heart for 8 hearts.
So the creative machine there where you attach the pumps, did you come up with that pump, or does that already exist?
Okay, these are very old pumps. The pumps themselves have been, these pumps are made by a company called Harvard Apparatus, and they've been around since about the 1970s. And the issue is that since they are analog, there's no way to actually really physically detect how their movement together. So I designed a circuit system that, since it's a pulsatile pump with an arm, that every time the arm moves forward, it closes a switch, which sends a piece of current to a little chip, which then sends that to my computer, which then is interpreted by the program to graphs with one positive for one pump, the other negative for the other pump, and that's all imported to an Excel file. Yeah.
Speckle to me means laser
Ultrasound, speckle. Yes. What was pumped through the heart, well, since it's a closed loop system with balloons that are actually, that replicate the function of the ventricles themselves, basically what's pumped through is a mixture of water and a little bit of cornstarch that simulates the blood flow, and that's what's the standard procedure that's used for passive studies.
More questions?
Oh, I'm sorry.
On your results chart, where you were giving the average percent strain, there you go. And you mentioned your 30 t-test and your standard deviation bars. How many standard deviations are represented by each of those bars?
So the question was how many standard deviations are represented by each of the bars. And what I did was that I averaged the data from each 0, 20, 40, 60, and 80. So there's just one standard deviation of the entire averaged, I compiled all the numbers into one data set. So each 0, 20, 40, 60, 80 is one standard deviation of the entire set of data for 0 milliseconds, 20 milliseconds, 40 milliseconds, and so on.
When you were looking at the video of the pumps that you were making, did you make those all yourself, or did you have help from people's labs?
Well, the question is how much did I make the setup myself versus how much input was at the lab. Basically, when I started out wanting to do this project, the head researcher of the lab said that I was crazy because they tried to do this before and it hadn't been done. And so they basically said, well, here's our pumps and here's how to suture and all this. And if you can figure out a way to make it work, then you can make it work. So I spent a lot of time working on trying to get this to work, and the entire setup was I created. Yes.
What kind of volume are you pumping? Is it comparable to the volume that the actual pigs are pumping? You know, if it was just a live normal, or are you pushing through a ton of volume?
Sorry. Well, the question was what exactly, how much volume I'm actually pushing through the hearts, is it, and how much you're correct, or is it not? And so the limit of these pumps for the stroke volume is 70 milliliters. And so I believe my study, the data I have here, the stroke volume was set at 60 milliliters. And normally the passive studies are done between 60 and 70 milliliters for the stroke volume.
Time's up.